which I claim is going to take fabrication in the way that Neil talked about, from analog to digital, and that's mainly what I'm here to talk about. First of all, Zymex Labs is a nanotechnology company, and I'm here to tell you that nanotechnology has not lived up to its promise.
Sorry, John, let me ask you to take a minute. Just give 1 minute on your own background. Who are you to be doing this?
You're wrecking my 5 minutes. I've been a maker my entire life. I'm an electrical engineer. Really, I'm a nano-fabricator, which might mean that I tell small lies. I wanted to thank you for bringing me here, because a month ago I had no idea that this fab lab process worked. Very exciting. Love the stuff you're doing. My father was an engineer. I'm in a mixed marriage. See, I'm an electrical engineer. My wife's a chemical engineer.
I mentioned this just because John has a long history in traditional chip fabrication and then realized we're going to crash and you have to get to the endpoint.
Absolutely. So one of the things that's preventing us from getting to the endpoint is the fact, again, nanofabrication not lived up to its promise. We have some great experiments in laboratories. We have a few simple products, but no complex systems. Where are the respire sites that are going to have 200 times the ability to carry oxygen in normal red blood cells? Where are the DNA sequencing nanopores? Where are the materials that are stronger than wood and lighter than air? Where are the single atom qubit devices? The reason that they haven't shown up there, in fact, is because our fabrication, all of our nanofab, ladies and gentlemen, is really analog. It's analog because it treats matter as if it's infinitely divisible. It largely ignores and usually does battle with the fact that matter is quantized. So I maintain that there's a digital revolution that's required here. So our approach is, at the atomic scale, is to take advantage not only of the chemical nature of atoms and molecules, but their wonderfully invariant spatial qualities to use that size control to control the size of the things we're fabricating. Our digital process, our binary process, is going to be making and breaking chemical bonds. It's going to require some process tolerance. Somebody asked me later about the tolerance of chemical bonds. It has to be digitally, and this separates us from chemistry, digitally, spatially address which chemical bonds are going to react with, and it must have error detection and correction, very important in a digital process. The one technology I'm going to talk about is a variation of e-beam lithography. It's the technology that most people use when they're doing experiments at the nanoscale, but ours is a digital process. It's done with a scanning tunneling microscope, and it's resist. The thing it's making patterns in is a single monolayer of hydrogen on a silicon surface. Now that needs to run. Somebody make that run. This is so disappointing. All right, this is supposed to show you how atoms are like people. They're not satisfied until all their needs are met, and their needs, in fact, are chemical bonds. In a three-dimensional structure, all the chemical bonds are satisfied. At the surface, they're not satisfied. They're unhappy. They're very reactive. We can bring in hydrogen atoms, passivate them, and using a scanning tunneling microscope tip, we can either just image where the atoms are, the hydrogen atoms on the silicon surface, or we can go into a litho mode and remove exactly the hydrogen atoms we want to remove.
So John, at a break, we'll download it separately and play it.
Sure. And so it allows us to put atoms where we remove the hydrogen atoms and build up three-dimensional structures. So this is a very digital process. I won't go into all the details, but any digital process buys you enormous tolerance. So compared to conventional analog lithography, we have an amazing, there's no overexposing the resistance. The hydrogen atom's gone, it's gone. There's no partial exposure of the resist. There's no proximity effect, which really affects dramatically normal e-beam lithography. And there's a spatial tolerance. It's small, but a manageable spatial tolerance is where we put the tip. So we have developed a tool that's commercially available. It's called a Zyvector. It turns an STM into this kind of patterning machine. It does sub-nanometer features on a regular basis.
Sorry, John,
and has atomic precision.
Those images are so interesting. Take a minute more to explain what's
All right, so up here on your left-hand side is a single, less than a nanometer lines of box where hydrogen's removed, little squares there, something that's supposed to look like circuitry. Up in here, actually, James Owen was an Oxford lab. That is a pattern that was made completely automated. It was actually a Celtic knot was on his wedding invitation, so he wanted to do that.
And the scale bar for that is 50 nanometers. A nanometer is maybe 10 atoms or so.
So let me help you
That's
out
a few
with
hundred
a nanometer, okay? And now I'm from Texas, so you got to put it this way. A nanometer is 1 billionth of a meter. Let's imagine the great state of Texas was 1 meter. Okay, 1 meter. How big would a nanometer be? When I ask people this, they say, well, maybe the city of Abilene. Actually, it's 4/100ths of 1 inch would be a nanometer. If the great state of Texas was a meter.
So his wedding invitation is a few hundred atoms plus.
Right, that's exactly right. So, this brings me to one of my major points, a new exponential trend that I'm calling the inverse Moore's Law. Moore's Law is about improving manufacturing precision to make smaller features, but to fit it on essentially a product that's been the same size for several decades. Okay? And it's what it does is been the same. It does information processing digitally. What I'm talking about is the inverse Moore's Law, where we stop improving manufacturing precision because we've reached the ultimate limit. It's absolute precision, but we're going to do that first at the nanoscale, some very valuable products at the nanoscale. Then we're going to scale up to ever larger products, ever more complex, dramatically, and because it can be digital, because you now have digital math, they can be extremely reliable, and we'll have this wonderful exponential growth to more and more complex materials, devices, and systems. The last thing I want to mention is if anybody wants to ask me about it, not during this panel, but later on, we're also working on healing the blind. So you can ask me about that if you're interested. I'd love to talk about that. That's what I wanted to talk about, ladies and gentlemen. Thank you very much.
So, when we wrote the book, Joel Allen and I fought over who got to use a William Gibson quote, the future is here today, it's just not uniformly distributed. And in this case, in John's lab, he's placing 1 atom at a time. You can do that now, and the question is just how quickly you roll that out. Next speaker is either a collaborator or a competitor. I'm not sure which, for the ability to do atomically precise manufacturing. Kate, and when you click that, sometimes you get sometimes you get 1 slide, sometimes you'll get 2 slides.
I don't have slides, I just have 1 video, so that's
Okay.
really easy.
So we can play Kate's video.
So, hi everyone, I'm Kate, I'm a bioengineer at University of Minnesota. And I will have a video, yes I do. What we do as biological engineers is we basically look at this incredibly complex machine that's called life cell. And we're trying to figure out how it works.
Lights on stage up please so we can see Kate.
Here. We're trying to figure out how this thing works, and more importantly, how can we make it to work. I can stand wherever I need. How can we make it to do what we want it to do? So we're using natural biodiversity. We're looking at live organisms that already do what we want to do. We analyze it and we take out specific genetic circuits, different single genes, to build new pathways, to build new genetic circuits that do exactly what we want to do. Make a new compound or make cells change in a certain way. And so once we have this new circuit, we want to dump it into another organism and analyze it, see how it works, get something out of it. And then we run into a problem, because cells kind of have a mind of their own, and natural biology has a lot of background. So when you put in a new circuit into an organism that already has a whole lot of its own circuits, you end up with a mess. You end up with those endogenous circuits interacting with whatever we made, whatever we put in there. And it's not only hard to see what's happening, but it's also hard to make anything happen, because there's interactions between what's already in the cell and what we put in. So what we do in our lab is we're trying to solve this problem. And we're basically trying to build a new, simpler chassis for biological engineering. We're working with those things called synthetic minimal cells, which are programmable, designable bioreactors that are made for a specific purpose, made with a very precise recipe to do whatever we want. So in a way, you can think of those synthetic cells as kind of biological machines. They're built to order, they're programmed to make two specific things, and there's no background of this messy biology running behind it. So all of the synthetic cells are built to a very specific recipe. So very briefly, how we make it is we start with an empty membrane, which is pretty much the same as a natural cellular membrane, And then one by one, we put elements. Those elements come from different cells. Those elements come from different source cells. And we end up with this synthetic cell that's basically a mosaic of properties of those different cells that we started with. And if you think of it as a biological machine, the DNA, the genes that we put in to program it, are like a program for that machine. And because the design is so modular, we can take elements, we can replace elements, and change the design basically on the go as we do the experiments. And to kind of give you the scale of the engineering problem we're having, humans have about 20,000 genes, bacteria have about order of magnitude less. The first live organism with a completely synthetic genome has 474 genes. And our synthetic cells have right now about 1,000 genes. So that's space between this non-living, completely synthetic cell with 1,000 genes and about where a live cell with a completely synthetic genome is right now is our sandbox, is where we do all of the prototyping, is where we can put our genes, put our new circuits. And when we make that synthetic cell, the reason to do it is because it's actually easier, cheaper, and faster than working with live cells. Because synthetic cells are not alive, you don't consider them genetically modified organisms, so there is no biosafety concerns. There are some, but much less than if you work with a live cell. We also, because we make them to order, we build them in the lab, we don't have to wait for a culture to grow. So the experiments, the iteration of each experiment is much faster. And because of all of those things, working with synthetic cells is much cheaper than working with natural cells. And as bioengineers, we consider ourselves engineers, and we do have a huge reproducibility problem. The traditional field working with live cells is dealing with this problem that no two live cells are ever alike. Even organisms that are genetically identical will end up just a tiny bit different because of all the changes, all of the epigenetic changes that happen during the lifetime. So when you measure any signal from live cells, you have just this ton of background that I talked about earlier, and the signal is extremely messy. But when you work with synthetic cells, all synthetic cells making one experiment end up pretty much identical. So the signal is extremely uniform. So if you want to program them, we're talking about manufacturing, if you want to program them to make a small molecule, or to make a peptide, or to make a protein assembly, it's much easier to detect what you've made and how that changes your design than if you have less background than if you're working with natural biology. And so basically you can make synthetic cells that produce toxic products, toxic small molecules or toxic proteins that would be very hard to make in natural cells. And you can make whatever product you want because you designed the synthetic cell. You design this cell so you work outside of the constraints of the natural biology. So in a way, you can think of synthetic cells as kind of a biological breadboard. You mix and match parts, and you can prototype your biological assembly that comes with basically only your design to constrain it. And so we use synthetic cells for other things too. We use it as a basic science tool. Some of the things we're doing is trying to figure out the origin of life, trying to figure out if there is life elsewhere in the universe. We're also using synthetic cells as a biomedical tool to do drug discovery and to figure out how diseases work, how diseases start, and how we cure them. But for the purpose of today's panel, biomanufacturing, we're using it basically as a programmable bioreactor that we can build